Welcome to Brain Ponderings. This will be the second in a 5-lecture series on neurobiology of aging. In the previous lecture, I talked about theories of aging, general theories of aging, comparative biology of aging, as well as the key animal models of aging that have been used to elucidate the underlying mechanisms. In the present lecture, I'm going to talk about the cellular molecular hallmarks of brain aging. And I'll be providing a lot of information pretty quickly. It's kind of an overview. So I'm not going to go into a lot of detail so that I can cover all the bases. This first slide covers the key hallmarks of brain aging that I'm going to talk about today at the cellular level. And these have been established through numerous studies over decades and decades of research on both animal models and humans. And humans, much of the data comes from postmortem histological analyses and biochemical and even molecular analyses. Now, one of the early theories of aging was the oxidative damage accumulation theory, which essentially said that over time, free radicals are constantly attacking molecules like DNA, proteins, membranes, and the damage accumulates. The cells are not able to repair it sufficiently to maintain the functionality and viability of the cells, and the cells uh, either develop a pathology or they die or they acquire certain inflammatory phenotypes, which I'll talk about. A second molecular hallmark or cellular hallmark of aging is dysfunction of mitochondria, the energy factory of the cell. And these mitochondria, their electron transport chain may not function well. They may generate a lot of free radicals and have damage to the various constituents. And related to this is impaired molecular waste disposal, that is to say, autophagy and other mechanisms of removing damaged cellular components. So, for example, in the case of dysfunctional mitochondria, there's a healthy way cells remove dysfunctional mitochondria by shuttling them to the lysosomes in a process called mitophagy. The other hallmark is impaired ability of cells to repair damage. So that could be impaired autophagy or impaired DNA repair. DNA repair is quite interesting because there's some very rare premature aging syndromes, the so-called progerias, in which mutations in genes encoding DNA repair enzymes cause this remarkable accelerated aging, such that individuals in their teens and 20s are looking like they're already in their 50s and 60s. And of course, they end up dying at an early age, usually what we would consider middle age. Uh, problem, two problems that are particularly relevant to the brain and the fact that neurons are excitable cells are aberrant neural network activity. So what I mean by that is a degradation of the ability of the neural networks to control their excitability within normal, useful, if you will, limits. And in particular, there's a hyperexcitability that occurs. So there seems to be problems in, for example, GABAergic inhibitory neurons controlling the excitability of glutamatergic neurons. during aging. Um, Related to that and damage and dysfunction of cells is an impaired ability of neurons to control calcium levels within physiological limits. And this problem is particularly uh, prominent at synapses, the sites where there's a lot of calcium influx when, uh, for example, glutamate activates postsynaptic receptors, but also in the presynaptic terminals. Um, Impaired adaptive stress response signaling. There are numerous signaling pathways that their function is to promote neural plasticity and survival. These include neurotrophic factors such as BDNF, NGF, um, GDNF, and others. These adaptive stress response pathways also include genes that encode antioxidant enzymes, protein chaperones that prevent protein misfolding, and others that I'll mention. Another problem which occurs during normal aging is that glial cells, and particularly microglia, become hyperactivated. They adopt a pro-inflammatory state. This is a state that is normally activated in response to a brain injury or an infection or something like that. But this can occur and does during normal aging. I should point out that all of these changes I'm talking about here are exacerbated in neurodegenerative disorders such as Alzheimer's, Parkinson's disease, frontotemporal dementia, and as well as in acute injuries like a stroke or a traumatic brain or spinal cord injury. Stem cell exhaustion, there is some evidence for this from animals that as they get older, the number of stem cells and neural stem cells in the hippocampus declines, and in particular their ability to generate neurons that are incorporated into the existing circuitry. Now, two other changes that occur during aging, one that's now just in the last 5 years or so been fairly well established, is some cells undergo senescence in brain aging, and in particular mitotic cells such as microglia, astrocytes, and oligodendrocyte progenitor cells. Telomere damage, there's less evidence for this in the brain. There's good evidence in mitotic cells that telomeres shorten during successive rounds of cell division during aging. In the previous lecture, I talked about how you can uh, culture cells, fibroblasts, skin fibroblasts from humans at different ages. And if you put them in culture from the skin of a young person, they will undergo many population doublings or replications before the telomeres shorten sufficiently to trigger senescence. Um, and so there is evidence that this can happen in cells that are dividing in the brain and neurons. Not much evidence. They're already post-mitotic. Okay, so I'm going to start out with the oxidative stress, where you have oxygen, which of course is used in mitochondria in the electron transport chain in the process of ATP production, the energy currency of cells. And during that process, particularly in mitochondrial complexes 1 and and also other mitochondrial complexes, a oxygen-free radical called superoxide anion radical is generated. And this can be converted through the activity of an enzyme called superoxide dismutase 2, SOD2, to hydrogen peroxide, which isn't a free radical, but uh, as you know, you can take hydrogen peroxide and it's a good antiseptic because uh, it can lead to the generation of free radicals that damage cells, and one of those is the hydroxyl radical. Um, and so these free radicals, these and others I'll mention, can lead to damage to membrane lipids and other lipids in cells, proteins, and nucleic acids, DNA and RNA. Um, there are repair mechanisms, but these may be compromised during aging. And also, there are intrinsic antioxidants. Uh, I mentioned one last time, actually, uric acid, but there are others. And so these normally help protect or get rid of these or repair the damage. And if the damage isn't repaired, it leads to cellular function decline and aging. Okay, so let's look at this oxidative stress pathways again. Superoxide anion radical in the mitochondria. Hydrogen peroxide can be converted to hydroxyl radical, and that occurs in the presence of ionic iron, Fe2 plus, or copper, Cu plus. So if you just take, for example, cultured neurons, um, and you add iron, ferrous iron to them, it can actually cause damage because of the endogenous hydrogen peroxide generated in the cell. And also this superoxide anion radical can interact with nitric oxide, which is a free radical uh, generated through the activity of an enzyme called nitric oxide synthase in neurons. And this occurs in response to calcium influx. So when a neuron fires, calcium goes in through membrane channels in the outer membrane, activates this enzyme, generates nitric oxide. Um, this nitric oxide serves normal functions. It has a signaling function in cells by um, activating cyclic GMP, cyclic GMP kinase pathway. However, this nitric oxide, sorry, can also interact with superoxide to generate this peroxynitrite free radical, which can cause membrane lipid peroxidation in a manner similar to hydroxyl radical. Okay, so lipid peroxidation is pretty interesting because neurons have in their membrane certain lipids such as arachidonic acid, a structure shown here that have multiple double bonds in the fatty acid carbon chains. And these bonds are prone to attack by hydroxyl radical and peroxynitrite. And when that happens, essentially the free radicals cause cleavage of this membrane lipid and resulting in the release or liberation of this product here called 4-hydroxynonenal, uh, a 9-carbon aldehyde. And this aldehyde can be toxic to cells because it can covalently modify proteins through this Michael addition process and cysteine, lysine, and histidine residues. And that can impair the function of the protein. And whether or not a protein is function is affected by this lipid peroxidation product 4-hydroxynonenal kind of depends on where the cysteines, lysines, histidines are and how critical they are to the normal function of the cell. But studies have shown that in neurons, some of the proteins that are rendered dysfunctional by lipid peroxidation are the membrane sodium pump and calcium pump. So the ion motive ATPases that move sodium and calcium out of the cell after the neuron has fired, the membranes depolarize. Glucose transporter, so this aldehyde can impair the ability of neurons to take up glucose and use it for ATP production. Glutamate transporters, the transporters in the membranes of neurons and astrocytes that move glutamate, remove it from the extracellular space so that it's no longer can bind to receptors. And that's a normal mechanism to prevent hyperexcitability, and that's compromised by this oxygen-free radical mediated lipid peroxidation process. There are ways you can look at levels of this aldehyde and their association with cells and even proteins. You can do uh, kind of a biochemical analysis. There's certain methods you can quantify this aldehyde. There's also antibodies that recognize proteins only when they're modified. That is, only when they have this aldehyde bound to them. So you can essentially immunostain cells under different conditions. Here's some cultured neurons under control conditions and after exposure to the amyloid beta peptide that forms the plaques in brain and Alzheimer's. So you see this black intensity here. That's the indication that this uh, Alzheimer's amyloid peptide is causing lipid peroxidation and the binding of this toxic aldehyde to proteins in the neurons. This shows the chemistry a little bit. So um, this is 4-hydroxynonenal bound to cysteine, lysine, or histidine residues. Also, with nitric oxide production, you can get protein nitration on tyrosine residues. It's Y is tyrosine. So that's shown here. And yeah, so there's been a lot of work on this. Interestingly, one of the main antioxidant molecules in pretty much all cells that's been looked at, including in the brain, is glutathione, which is just a 3 amino acid molecule with glutamate, cysteine, and glycine. And it turns out that if cells are exposed to conditions that cause lipid peroxidation, this glutathione sops up, if you will, a lot of the 4-hydroxynonenal because it binds to the cysteine. And then the cells actually, they have some mechanism that recognizes this aldehyde bound to the glutathione, and the glutathione is excreted from the cells. It's ejected essentially from the cells. So this seems to be an important way cells protect themselves from excessive, uh, the adverse effects of excessive lipid peroxidation. Now, it turns out you can see at a subcellular level indicators of the accumulation of oxidatively modified proteins in neurons and other cells. And so, for example, there's two really well-established examples. This is an electron micrograph of dopaminergic neurons in the brain of a 78-year-old human who had passed away, and then it's at autopsy, the tissue was prepared in thin sections to look under the electron microscope. And what you see is these black electron-dense uh, uh, blobs, if you will, here. And these are neuromelanin, which is it's kind of similar to melanin in the skin that can make the skin black, have black color and absorb light, but it's not melanin itself. It's this dihydroxyindole, and there's polymers of it, and accumulates as a result of oxidative stress in these dopaminergic neurons. This just during normal aging, this person hadn't been diagnosed with Parkinson's. This occurs to a much bigger extent in Parkinson's. The other uh, you can actually see another oxidative stress marker under EM, and that's so-called lipofuscin, which is fluorescent under ultraviolet light, interestingly. So even in tissue sections that you would use like to do histology for immunostaining, whatever, and if you do that on an unstained brain section from a very old human, you'll see fluorescence spots, and those are accumulation of oxidatively modified lipids. And this just shows that there's a progressive increase in uh, neuromelanin accumulation in substantia nigra with age. Um, a marker of nitric oxide and peroxynitrate activity increase is using nitrotyrosine antibodies, which recognize the this modification here, nitration on the tyrosine residues. And there's antibodies that have been developed that recognize nitrotyrosine, and this is staining of an elderly Alzheimer's patient, showing that neurofibrillary tangles have a lot of nitrotyrosine in those. So these are degenerating neurons in Alzheimer's. This looks at mitochondria in a little more detail, just kind of repeating what I already said. Superoxide is generated through the normal activity of complexes 1 and 3. It's normally eliminated by manganese SOD or SOD2, converted to hydrogen peroxide. Hydrogen peroxide is by itself not a free radical, but in the presence of iron and copper can generate hydroxyl radical and cause a lot of damage to DNA, membrane lipids, and other molecules. There are enzymes that normally get rid of hydrogen peroxide. These are glutathione peroxidase and catalase. A few other things on the mitochondria just, that are interesting from the standpoint of neurons. These mitochondria can take up calcium. So when a neuron fires and you get calcium influx, some of that calcium is taken up by the mitochondria. It actually stimulates an increase in activity in the electron transport chain to generate more ATP, which is often needed when a neuron fires to run the sodium and calcium pumps that restore the ion gradients. But when a neuron fires and the calcium goes into the mitochondria, it also increases the generation of superoxide and potentially free radical production and damage. Uh, Another interesting thing is mitochondria play a very important role in a cell's decision, if you will, to die or not to die. So to undergo programmed cell death, apoptosis or not. And so the mitochondria, there's certain conditions where it's not so much in neurons, but certainly in other cell types that can be easily replaced from stem cells. It's better to get rid of a damaged cell by apoptosis than have it continue to survive and undergo necrosis where the membranes break and it spills its toxic contents that could adversely affect other cells. But anyway, that's a whole nother lecture. Okay, so during development, neurons are born from stem cells. They extend several neurites. One of them starts to grow fast. It becomes the axon. The other ones branch and grow and they become dendrites. And during this process, the number of mitochondria increase a lot, and those mitochondria are moved out the axon dendrites so that they can carry out their functions at sites where they're needed, such as synapses, in or near synapses, where there's a big demand for ATP. And also, as I mentioned, the mitochondria play an important role in calcium regulation in neurons. Yeah, and so as synapses form mitochondria, there are mitochondria in the presynaptic terminal. You don't really see them in postsynaptic dendritic spines, but you do see them right at the base of the spines. I'll show some examples of that from electron micrographs. This lower left images are of a cultured embryonic rat hippocampal neuron at 3 days in culture. And these are stained with a fluorescent probe for mitochondria that's shown in red here. So during this 1 week period between days 3 and 10, you see a big increase in the number of mitochondria, and they fill the dendrites. And this lower right just shows that there are molecular mechanisms that move mitochondria within neurons, and essentially they move along mitochondria, and there are these energy-dependent motor proteins, kinesins, and dyneins that move them in one direction or another. I went into this process in detail in my one of my previous lectures in my series on brain ponderings in brain bioenergetics. Okay, this is an example from Pamela Liao and Tom Delgado in my lab, where they essentially uh, did 3D reconstructions uh, from electron microscope images. So the way this is done, hundreds of very thin, essentially close to two-dimensional images are acquired, and then through the power of the computer, they can reconstruct and get a 3D image. And so what you see here is an example of a dendrite here with one long mitochondria extending through this region of the dendrite, that's shown in pink here. And you have several presynaptic terminals, which are this light yellow here. And you see the mitochondria in this particular image in two of those presynaptic terminals, but you don't see any mitochondria in the dendritic spine shown here. So that's what I mentioned before. Okay, I mentioned earlier mitochondria can divide. Okay, so that's mitochondrial biogenesis that occurs dramatically during development and formation of neural circuits, but it's also constantly ongoing because some mitochondria will invariably become dysfunctional and they're eliminated by mitophagy, moving to the lysosomes. And healthy mitochondria or even parts of mitochondria divide. And I'm not going to go into detail on this. Again, I did that in my brain bioenergetics series, but the transcription factors and signals that stimulate mitochondrial biogenesis have been in part identified. So some of the stimuli that will lead to increased number of mitochondria in neurons and muscle cells is exercise, physical exercise, also cognitive stimulation. Just when neurons are active, that's actually a signal that the cell is experiencing a lot of high energy demand and stress, and so we better increase the number of mitochondria, and also actually at the same time stimulate mitophagy to get rid of dysfunctional mitochondria. In the last lecture in the present series on neurobiology of aging, I'm going to come back to this in the context of interventions that can counteract brain aging, among which caloric restriction and fasting and exercise and cognitive stimulation with recovery periods in between these challenges seems to be good counteract aging. All right, I also wanted to mention here interesting class of enzymes called protein deacylases or deacetylases. And these were really initially discovered to be important for aging by work in yeast from Len Guarante's lab. where they identified a gene called SIR2, SIR2, that that seems to be involved in the aging of yeast. And if you increase its levels, SIR2 levels, it seems to slow down aging. If you knock out the gene, it's accelerated aging. And there's some evidence that the mammalian homolog SIRT1, SIR2 and 1, may also play some role in counteracting aging. There's also interesting relationships between SIRT1 and some of these other protein deacetylases and cellular stress responses and cellular energy metabolism. In mitochondria of cells, including neurons, the SIRT3 seems to be particularly important. It has a number of different protein substrates. And so what this enzyme does is it's a deacetylase. It removes acetyl groups from lysine, the amino acid lysine in proteins. So it's a lysine deacetylase. There are other enzymes that can add acetyl groups to the same lysines. These are lysine acetyltransferases, or uh, they're acetyltransferases. Okay, so there's at least several hundred proteins in mitochondria that have been shown to be deacetylated by SIRT3. These include SOD2, this very important antioxidant enzyme in the mitochondria, uh, proteins involved in the TCA cycle and electron transport chain, so involved in ATP production, fatty acid metabolism, uh, translation, protein translation, interestingly, transporters, and proteins that regulate mitochondrial DNA replication. I'll come back to that in a subsequent lecture in this series, some very interesting studies in which SIRT3 levels have been manipulated in mice and shown to have really interesting effects on synaptic plasticity, neuronal vulnerability to stress and disease. Okay, autophagy, very important process whereby cells recognize and eliminate damaged proteins, organelles, membranes of organelles, uh, and other molecules. And the way this works at a simplistic level is cells have proteins that are capable of recognizing, for example, misfolded or oxidatively damaged proteins and then shuttling them to the lysosome. So the red here is lysosomes. And also organelles or membranes can be identified. So here this shows mitochondria are actually more commonly part of a mitochondria. These mitochondria actually often bleb out, and when there's part of the mitochondria that's damaged, that part is released by essentially blebbing and release of vesicles containing the damaged constituents, and then they're recognized through this molecular machinery and form these autophagic vacuoles that are then shuttled to the lysosomes, and the contents are released, integrated in the lysosomes. There's also a process called chaperone-mediated autophagy that mainly targets misfolded and oxidatively modified proteins, or perhaps protein aggregates such as those that accumulate in the brain in pretty much every major neurodegenerative disorder: Alzheimer's, Parkinson's, Huntington's, ALS, frontotemporal dementia, etc. I'll talk about that in a subsequent lecture. A lot of interesting work's been done on trying to understand how lysosomes function and what regulates their function. The pH of the lysosome inside the lysosome is very low. It's acidic, and there are enzymes in the in the lysosomes that their activity functions very well at this low pH. And these enzymes degrade cargoes that are coming in from damaged proteins or organelles. The lysosome also has regulatory systems for calcium. There's channels, for example, and calcium flux across the the lysosomal membrane can affect the enzymatic activity and functionality of the lysosomes. In fact, there's some evidence that in aging and Alzheimer's disease, the pH in the lysosomes increases and lysosome function is impaired, and that can lead to a buildup of damaged molecules and organelles that would otherwise be degraded in the lysosome. So this is perhaps one way that the process starts during aging that leads to diseases where you get these proteins and organelles accumulation in Alzheimer's, Parkinson's, etc. Now there's a number of ways to look at this state of autophagy and more increasingly more methods. This kind of a neat method where they developed this construct where you have a red fluorescent protein, green fluorescent protein coupled to LC3, which is a protein involved in autophagy. And in this particular study, uh, they used a Thi1 promoter to make mice that express this construct in cells and neurons in particular. And then, so what happens then is when you look at the cells under the microscope, if the lysosomes are functioning well, then you'll get red fluorescence. If they're not functioning well, you won't get red fluorescence. You're going to get more yellowish fluorescence. So this just shows some uh, neurons in the brains of control conditions, a lot of red, healthy functioning lysosomes, and uh, and this just shows where you add chloroquine, which raises the lysosomal pH. They're not functioning well, and they're no longer red. Um, so I mentioned that mitochondria can control cell death survival processes through these pathways. I'm not going to go into the detail. There's a lot of proteins like tumor suppressor proteins, P53. The reason that mutations in P53 are associated with cancer is because one of its normal functions is to P53 increases when you get accumulation of DNA damage in cells and they can potentially become cancerous. So P53 goes up and that triggers death of that potentially cancerous cell. So that's the normal way that we eliminate potentially cancerous cells. And if you have a mutation in P53, which a lot of cancers do, then this normal mechanism for eliminating that potentially cancerous cell is compromised and they're not eliminated in cancerous form. And there's also a relationship between mitophagy and apoptosis, so that if mitophagy is impaired to sufficient extent, it can trigger death of cells. And if that happens in neurons, that's not good because they cannot, for the most part, be replaced. And, so I'll talk about in a couple lectures about some of these proteins that are involved in mitophagy and how mutations in them can cause early onset Parkinson's disease. Uh, on the other hand, there are some of these other pathways, the sirtuins uh, and AMP kinase, and their effects, for example, through mTOR can can protect against pathology. Uh, Now, the mTOR pathway, it's a cell growth pathway. It's a pathway by which cells move nutrients into the cell, particularly amino acids, and then use those amino acids to synthesize proteins. So when the mTOR pathway is highly active, cells are actively synthesizing many, many, many hundreds, thousands of proteins, and they can grow and undergo plasticity, for example, synaptic plasticity, neurogenesis. So you have to have mTOR pathway activation for that. But constant excessive activation of the mTOR pathway uh, inhibits autophagy. And so, for example, if you overnourish cells, such as in many humans nowadays with obesity, you will get chronic activation of the mTOR pathway, this kind of cell growth pathway, and autophagy is impaired and it accelerates in that way disease. In the fifth lecture in this series, I'm going to talk a lot about the importance of switching back and forth between a cellular growth state and a recovery period, and sorry, between a stress, if you will, challenge period when nutrient levels are relatively low. So that's like during fasting, extended periods between meals with vigorous exercise, using neurons actively. So that, as I'll talk about later in my last lecture, those conditions are actually the cells, neurons perceive those as a mild stress, bioenergetic stress, and they respond adaptively by, on the one hand, inhibiting mTOR pathway, it's down, and activating autophagy, activating, for example, DNA repair pathways or production of protein chaperones that prevent protein misfolding, and then switching between that cell stress resistance, conserve resources mode and the growth and plasticity mode uh, seems to optimize health, including brain health. So stay tuned. DNA repair. A lot of work been done on this, identifying the proteins that control DNA repair, very elegant systems cells have for detecting and and repairing DNA that's been damaged by free radicals, Nobel Prize given 10 years ago for that. I'm going to focus on, and there's multiple mechanisms of DNA repair, but only a few, maybe one or two, a couple actually, apply to neurons, which are postmitotic cells. The other mechanisms apply to mitotic cells and require DNA replication. All right, so as I mentioned, DNA can be damaged by free radicals, and the guanine base in DNA is particularly prone to attack by a hydroxyl radical, for example. And with aging, the accumulation of oxidatively modified DNA increases in cells in the brain, including in neurons. And and there are many things that can lead to free radical production, but the main thing is actually, and DNA damage is oxidative stress, this from the normal activity of the cells and mitochondrial activity. But other things that can cause DNA damage is ultraviolet light can cause mutations. Think skin cancer. Certain chemicals can cause DNA damage, uh, DNA crosslinks, and so on. Um, and accumulation of DNA damage can lead to certain disorders specific to DNA damage, but more non-specific things and may play a role in brain aging and neurodegeneration. Uh, I'm going to focus on this base excision repair, and there's several different variations on this, but I'm going to focus on just kind of the main pathway that we think is occurring in neurons. Yeah, I should, I kind of pointed out this before that if you have good DNA repair, um, cells can stay viable, functional. They're not going to undergo apoptosis, for example. You get too much DNA damage, cells may die, that may become dysfunctional or undergo senescence. I'm going to talk about senescence later. I briefly mentioned apoptosis, programmed cell death, which can be beneficial. Uh, it can also be detrimental. It may occur somewhat in neurodegenerative disorders or some variation on apoptosis. For example, I talked all about iron being important for generation of um, hydroxyl radical from hydrogen peroxide and then consequently attack of the double bonds in the membrane lipids by the hydroxyl radical and 4-hydroxynonenal, a toxic lipid peroxidation product. Actually, we did a lot of work in my lab on that in the 90s, and others did, the 4-hydroxynonenal iron story. And more recently, some other lab came up with a fancy term for that called ferroptosis, iron slash lipid peroxidation mediated cell death. Um, but in some instances, accumulation of damage may lead to the cells becoming senescent, and this particularly studied in mitotic cells in which they stop dividing, they become larger and start excreting a lot of pro-inflammatory cytokines, which lead to inflammation. I just want to mention briefly, I'm not going to go into any detail at all. There are these premature aging syndromes that involve mutations in certain DNA repair enzymes. Um, in this case, these are involved in repair of double strand breaks, but there are others that can lead to uh, impaired um, base excision repair. So this is an example from one of these premature aging syndromes where the brain's been looked at probably the most of any of them. And so these people, individuals, when they're in their 20s and 30s, they have cognitive and other psychological problems. If you look at their brains, there's abnormalities at the structural and also metabolic levels. And in general, they have accelerated brain aging. In other words, if you look at their brain at autopsy, it 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 can look like a age in normal individuals, although there's some still controversy on that. The problem with studying these subjects is there's very few of them. They're so rare, less than 1 in 100,000, that any of these studies typically only have a few, two or three brains to look at, and it's hard to make any firm conclusions. You can't really do statistics on it. Okay, this is the basic excision repair pathway that's so important in neurons. There's a when you get free radical modification attack on guanine in the DNA, there's DNA glycosylase that recognizes that modified DNA base, and then there's a enzyme AP endonuclease that removes the damaged DNA base, and then there's DNA polymerase beta that then uh, puts in, if you will, re fills in that gap with an undamaged DNA base. And it's been shown that This basic excision repair pathway declines. Its levels of functionality decline in brain cells during aging. It's been also shown um, that there's even a further decrease in basic excision repair ability in humans with mild cognitive impairment or Alzheimer's disease. And then there's been studies manipulating some of these enzymes in mice, in mouse models that provide evidence that if you compromise this basic excision repair pathway, it can, for example, accelerate pathology and cognitive decline in mouse models of Alzheimer's disease. Okay, now I mentioned neural network hyperexcitability as a feature of aging. Um, During normal aging, the incidence of epileptic seizures increases, particularly in late in 60s, 70s, 80s. Uh, Individuals diagnosed with Alzheimer's disease have up to a tenfold increase in seizure incidence, and if you record EEG from their brains, they often have, compared to age-matched controls, the Alzheimer's or mild cognitive impairment patients have um, these bursts of hyperactivity of neural circuits that are what are called subclinical. That the patients don't experience seizures, but nevertheless, there's this hyperexcitability detected. That's been shown also in animal models of uh, Alzheimer's. Okay, the main circuitry in the brain throughout the brain, cerebral cortex, hippocampus, basal ganglia, cerebellum, is excitatory glutamatergic neurons. About 90% of the neurons in the brain are glutamatergic. About 8, 9% are inhibitory GABAergic neurons. These glutamatergic neurons are often very large axons that can extend relatively long distances within and between regions, brain regions, or even between hemispheres of the brain, or even in the case of upper motor neurons down into the spinal cord. Um, and these GABAergic neurons are smaller and their axons don't extend long distances. They essentially act locally to when a glutamatergic neuron fires, these, I guess I didn't show that in this. Yeah, I showed it here in this diagram. This GABAergic neuron synapses on a glutamatergic neuron. So when the neurons fire in this circuit, this inhibitory neuron is activated, and then it's kind of a rapid negative feedback to quiet the neuron and the glutamatergic neurons and prepare it for another incoming input. Now, last thing I'll say about this slide is that all of our behaviors, everything we do, think, um, feel, if you will, is based on activity of these glutamatergic neurons. The only way any other neurotransmitters affect our behavior, whether they're dopaminergic, GABAergic, serotonergic, cholinergic, the only way these other neurotransmitters, dopamine, serotonin, etc., affect our behavior and function, mood, etc., is by modifying the ongoing activity of these glutamatergic neurons. And this is what had a large part of my book, Sculptor and Destroyer, on glutamate, the brain's most important neurotransmitter, was about. So if you're interested in understanding the brain, you better know about glutamate, and my book's not a bad place to start. Okay, this is just kind of a review up to this point. Aging, you get these changes that are occurring, and this can lead to impairment of regulation of neural network excitability, for example, through oxidative stress, uh, uh, accumulation of dysfunctional mitochondria, etc. And this leads to neural network hyperexcitability and can lead to a process called excitotoxicity when the neurons are actually physically degenerate. as the result of excess uh, activation of glutamate receptors. And that involves excessive calcium influx and accumulation in the neurons, mitochondrial dysfunction. This just shows what I mentioned, that seizures increase in normal people during aging. You'll see here, some of you may know this if you've had children or uh, have relatives that have had this with their children. Children just in the early years after they're born may have seizures, but most commonly they disappear by the time they're 3, 4, 5 years old, and they never have any seizures again. Uh, but later in life, people can develop seizures. And then with Alzheimer's disease, increased incidence of seizures, same with mild cognitive impairment compared to healthy controls. Okay, just quickly, excitotoxicity, I wanted to mention that so most of the synapses in the brain are glutamatergic. You have glutamate being released from presynaptic terminals and vesicles. When that presynaptic neuron fires, you get calcium influx in the presynaptic terminal. Glutamate's released. It can bind to several different type of receptors. I'm showing three, uh, well, yeah, three major types here. The AMPA and kainate receptors, which are sodium channels mainly and depolarize the cell. And then glutamate also binds to the NMDA receptor, which is a calcium channel. So glutamate causes membrane depolarization by activating these receptors, AMPA kainate, and then that depolarizes the membrane, and then glutamate can open this NMDA receptor channel. You get calcium influx. Calcium has many important functions in neurons, both in regulating the function of synapses acutely, for example, by activating protein kinases that transfer a phosphate group to target proteins, for example, ion channels, but also various receptors, other protein targets in the cells. Calcium also has very important, more delayed roles in neuroplasticity, for example, by activating the transcription factor CREB, which I'll mention later. But here, this just shows that excessive calcium can can lead through a number of pathways to damage to neurons. And this just shows some of the ways in which oxidative stress and calcium can, uh, if they're excessive and not dealt with by removing the free radicals or repairing the damage, or by removing calcium from the cytoplasm after the influx, uh, can lead to cytotoxicity. There's a number of ways one can protect neurons, at least in culture and in some cases in animal models, against excitotoxicity. The problem with manipulating glutamate system is that it's so important for learning and memory and all our behaviors that pretty much any drug that has any major impact on the glutamate system or calcium system is going to interfere with normal brain function. Having said that, there is one drug, memantine, that's prescribed to patients with early Alzheimer's disease. It's an NMDA receptor channel open blocker, so it gets into this channel and can inhibit calcium influx. And that has a small but significant effect in slowing down the decline. On the other hand, if you get too high dose of memantine, it can cause psychotic like behavior, kind of like you see with ketamine and psychedelics, actually. There are other approaches, though, for example, neurotrophic factors. And this is important when I get to my last talk, because it turns out that things like physical exercise, cognitive stimulation, like we're doing now, these physiological good stresses, as long as you have a recovery period, they upregulate these neurotrophic factors and uh, which we think play some important role in their benefits on neuroplasticity and protecting against hyperexcitability, for example. Um, yeah, so this is just more detail. I'm not going to go into details. This is from an article, yeah, I wrote a long time ago, actually, on role of calcium in brain aging and Alzheimer's disease and mechanisms and ways to protect against that. Okay, so what haven't I covered yet? I haven't covered inflammation, I haven't covered senescence, and I haven't covered telomeres and telomerase. Inflammation in the brain, one of the main sources of local innate immune system activation is the microglia, the brain equivalent of macrophages, these cells that can recognize damaged cells or accumulation of some damage and as well as infectious agents uh, to some extent and remove them by phagocytosis. Microglia have normal functions in the brain. They're not just sitting around during your lifetime waiting for some damage to occur or some infection to occur. They play important roles in kind of subtly uh, modifying synaptic plasticity. For example, there's evidence that these microglia can remove dysfunctional or synapses and eliminate them without adversely affecting the cell. So that's the so-called M2-like phenotype. They play normal functions. Um, they also may produce some neurotrophic factors. Then there's this M1-like activation state of the microglia where they generate a lot of pro-inflammatory cytokines like tumor necrosis factor, for example, and also reactive oxygen species, nitric oxide, or even uh, other reactive oxygen species. Um, if this occurs transiently, it may play an important role in removing debris because these can remove debris. But if it occurs chronically over periods of weeks, months, years, this is thought to contribute to damage to neurons through uh, kind of classic inflammation, chronic inflammation pathways, which involve oxidative stress and some of the other age-related processes I talked about. Okay, so cell senescence. Cell senescence is defined classically as in mitotic cells, a cell stops dividing, it isn't eliminated by apoptosis, and it often gets bigger in size and starts spitting out a lot of inflammatory molecules. And there's been some nice studies now showing that there are ways to eliminate senescent cells from tissues and through genetic mechanisms. I'll talk about one or two key uh, proteins involved in this transition uh, from normal cell to a senescent cell. And one of those is P21, another is called P16. So there have been mice where, for example, you can manipulate P16 up or down. If you knock down or out P16, then um, it'll prevent and even eliminate senescent cells that are already present in the tissue. And that seems to has an anti-aging effect in the tissues it's been looked at. I'll briefly mention that. In the brain, the question is, this is a recent revered article, I think, from Darren Baker's group up at the Mayo Clinic in my hometown of Rochester, Minnesota. And they had evidence, and all of this is really recent in the last 5 years that, or a little bit more than that, 7 years, that microglia Some of them may become senescent and maybe astrocytes. And we had evidence that oligodendrocyte progenitor cells uh, can become senescent with aging, and particularly in our Alzheimer's models. And not only that, but if you prevent the accumulation and eliminate these senescent oligodendrocyte progenitors or microglia that are spitting out a lot of pro-inflammatory cytokines, it can ameliorate the cognitive impairment and lessen somewhat the neuropathology in these mouse models of Alzheimer's. And I think I'll mention that today. Okay, so this is looks at a little more. If you get excessive accumulation of damage in cells, they can be eliminated by apoptosis, which can be good, at least in mitotic cells. But if they're not eliminated, they can undergo cell cycle arrest and acquire this senescent phenotype. Now, these cells, senescent cells, are resistant to apoptosis, but they're also, and there's evidence from the late Judy Campisi and others that they can contribute to cancer. Not necessarily that this cell becomes cancerous, but that if there's cancer forming, particularly during aging, which is when most commonly occurs, and there's senescent cells around, it can these senescent cells can kind of help the cancer cells survive and proliferate. Um, right. Uh, there's some evidence the immune system can be activated to clear to clear senescent cells. That seems to be compromised during normal aging. And again, these don't undergo apoptosis. Right. This just shows another look at this, focusing on when these cells are senescent, they're spitting out superoxide, nitric oxide, pro-inflammatory cytokines, and these senescent cells have increased P16 ink and P21. And there's been, so that's one way to eliminate this is genetically, but that's not so easy to do in vivo, certainly in humans. So there's one cocktail of two chemicals that have been shown to eliminate senescent cells. And that involves quercetin, which is actually a natural product that's present in the, for example, skin of apples and other fruits and vegetables. So quercetin in combination with a cancer drug called dasatinib. So dasatinib plus quercetin. And Pei Su Zhang, who unfortunately passed away a few years ago um, uh, of a brain aneurysm, she, in my lab, she used the 3xTG mouse model of Alzheimer's disease, which is a model developed in Frank LaFurla's lab with our help. We provided some mice that enabled him to do this, the presenilin 1 mutant uh, knock-in mice. And so anyway, the bottom line is these 3xTGAD mice, they express three different gene mutations, one in presenilin 1, which is an enzyme that cleaves the amyloid precursor protein to liberate the amyloid beta peptide that forms the plaques in the brain. And then the other two genes are tau, a mutant form of tau that causes frontotemporal dementia, and then also another APP mutation. And so these mice, as they age, they develop amyloid plaques, intracellular tau, neurofibrillary tangle-like pathology. They develop cognitive impairment. Um, and so in these mice, when we give them this, what we'll call senolytic, or this cocktail of drugs that removes senescent cells, we find that that improves their cognitive function. That is, so in this case, this is a normal learning curve. Uh, not the normal learning curve. This is all Alzheimer's mice. Uh, this gray is the learning curve for the Alzheimer's mice without removing senescent cells, and this is with removing the senescent cells. So the learning curve is steeper. They're more quickly learning this maze test, and this shows some example here. They're also better at remembering where, uh, how to solve this maze, if you will. And we found in this study that the accumulation of senescent oligodendrocyte progenitor cells in the plaque environment is associated with the cognitive impairment if we remove those cells. It ameliorates the cognitive deficits. These two graphs here are data from actually humans, non-demented controls, mild cognitive impairment, and Alzheimer's disease, looking at number of plaques and number of plaques where there are cells that uh, have a oligodendrocyte progenitor cell phenotype, which we find have also senescent phenotype. Okay, lastly, the telomeres, the ends of chromosomes. There's a 6-base DNA repeat that the telomerase enzyme called TERT, which is a reverse transcriptase, it adds this 6-base DNA repeat TTA GGG onto the ends of chromosomes. In mitotic cells undergoing cell divisions, uh, this enzyme is critical in preventing the shortening of the telomeres, the chromosome ends, during successive rounds of cell division. If you don't have this enzyme, um, the telomeres will get shorter and it will trigger cell death or other problems. And it's been shown during normal aging, however, that the telomeres still do get shorter. This is just a chromosome spread here of the human chromosomes with the yellow labeling the telomeres, the chromosome ends with a particular marker. Right. All right. So now that's all and good in mitotic cells, but neurons don't divide by definition. And individual neurons stay around in your brain for your entire lifespan after they're initially generated and survive the initial period of programmed cell death during brain development. Okay, so then the question is, okay, they're not dividing. Are their telomeres getting shorter? There's no convincing evidence for that. Uh, is there otherwise telomere damage? Uh, Possibly, because you have GGG, right? So if you remember from previous slide, I mentioned that these guanines seem to be particularly prone to oxidative modification. So you might predict that the telomeres would be prone to oxidative damage. And if that's not repaired, that could be a problem. Um, there's been a lot of work showing and these are in white blood cells, essentially looking at age and uh, telomere length, essentially. So telomere length declines in many mitotic tissues during aging, and that's fairly consistent. Although there's a lot of scatter here, right? In other words, there's a little bit of overlap here. Sorry, a lot of scattered variability among individuals, which makes sense. There's variability among individuals in their relative rate of aging and development of age-related diseases depending on their genetics, their environment, their lifestyle, their habits. Uh, right. Interestingly, though, we had shown uh, quite a while ago, this is 24 years ago. Let me go back to this. I just want to point something out here, right? So we did a lot of work in cell cultures where we got post-mitotic neurons and we're exposing them to amyloid beta peptide or causing excitotoxicity with excessive glutamate or causing metabolic stress by impairing mitochondrial function and looking at the mechanisms of cell death and what measures can our signaling pathways can protect against such cell death, such as neurotrophic factor signaling pathways, et cetera. And we were interested in brain aging, of course, and we knew that all the work at telomerase up till then had been in mitotic cells. So we started looking at this in our cultured neurons. And um, and what we found is that if you first in vivo, if you just look at the TERT levels, the telomerase reverse transcriptase levels by aminoblot, very high levels during embryonic development when cells are mitotic, and then uh, between P5 and P10, postnatal day 10, there's a dramatic decline, and that pretty much coincides with uh, a decrease in proliferation of glial cells, actually, which the glial cells are proliferated and born after the neurons are already there. Um, but there still is some TERT level, um, and this is in the hippocampus, so this cortex and hippocampus. And then we looked in at telomerase activity, and it decreases in somewhat lockstep with the decreased mTOR, although not exactly. So that's in vivo. And then we, in these culture neurons, if we reduce TERT levels by uh, using an antisense technology, which has been, since we did this work, this was like the cutting edge way to uh, knock out or knock down gene expression with antisense constructs. Uh, but what we found was that when we reduced TERT levels in these neurons, few of them survive over time, and particularly when we expose them to the amyloid beta peptides. So the bottom line is that we knock down TERT, the neurons are more vulnerable to being killed by not only the amyloid beta peptide, but we also looked at glutamate toxicity. So we wondered what was going on here. You know, is there some function of TERT other than being a reverse transcriptase? And we had some other evidence that indeed this neuroprotective mechanism of TERT in these post-mitotic neurons seemed to be independent of the reverse transcriptase activity. And um, and since then, Rhonda Pino's lab down at Baylor has uh, used genetic approaches in mice to to manipulate TERT and essentially uh, in a way that he can parse out any effect mediated by uh, reverse transcriptase activation and lengthening of telomeres or prevention of shortening from telomeres from some other activity or function of TERT. And his evidence, which is in this Nature Aging paper, if you're interested, very nice paper in 2021, what they've essentially shown through a number of really nice experiments is that TERT acts as a transcriptional regulator, so this is completely independent of its effects on telomeres, to upregulate the expression of genes such as brain-derived neurotrophic factor HSP70, which we and others actually had shown that BDNF, HSP70, and other protein chaperones can protect neurons against the adversities of aging, things like oxidative stress, metabolic stress, excitotoxic stress. And he showed through this mechanism um, the TERT can protect neurons against A beta. So that was kind of nice. 20 years later, uh, they confirmed our findings in cell culture. Okay, so that's the end of this lecture. Next time, I'm going to it's kind of be a hybrid lecture. I'll talk some about functional decline of the brain during aging, what kinds of functional abilities decline with what trajectories, structural changes in the brain, uh, MRI data, both looking at size of different brain regions and gray matter versus white matter, and a little bit on functional MRI connectivity, that is communication between brain regions and living human subjects, and some similar things from animal studies looking at number of synapses, et cetera, neurogenesis, things like that during normal aging. And I'm also going to talk a little bit about um, the mTOR pathway a little more because there's some interesting work done recently on amino acid restriction, dietary restriction, and effects on brain aging. So thanks for your attention, and until the next lecture. Goodbye.